Folks in the back. Our McGraw-Hill rep, Peter, has noted that a lot of you have courtesy access to McGraw-Hill that's about to expire. So if that's true, if you're using the trial period and it's about to expire, he wanted me to send you this one screenshot, how to upgrade to the full Connect package by buying online if you want. The bookstore, again, as I said last time, is expected to have more of the loose leaf plus Connect codes in by today. So if you're still waiting, please go there. Make sure that you get access to LearnSmart some way or another, because without it and without doing the online reading, there is not a very good chance of success in this class. Okay. So I've got a question for you about one piece of terminology that we haven't encountered yet in class, and that's what an isochore is. So here's a new term. You haven't read about this in the book, but this is pretty simple. When we're talking about Chargaff's rule, AT content, GC content, there's a really simple way to plot an isochore. So on one axis, we're going to have the percent G and C. That's the y-axis. And as is depicted here already, the x-axis is some DNA sequence. So essentially what an isochore plot is it's a moving plot of what's the percent GC in small little bits of sequence as you move down a sequence from the 5' end to the 3' end. So let's say in this case that we're making an isochore plot when we're looking at 5 nucleotides at a time. So this first window, what we call it, window 1, what's the percent GC in those first 5 nucleotides? Out of 5, there's a G and a C, so 2 out of 5, 40%. So we've got 50% GC, 100% GC, so that first window is at something like 40%. What about window 2? What's the percent GC in those 5 nucleotides? Pardon? Same, still 40%. There are 2 Gs out of 5 nucleotides. What happens next? We have 60%, 3 Cs out of 5, and then 80%, 4 Gs out of 5 in the 4th window, and then back down to 40%. So that's an isochore plot of this particular DNA sequence. What does it tell us? It's just a simple graphical representation of how many Gs and Cs there are at different points along a chromosome. And the reason I'm telling you this is because you've been voting on which organism you think has a higher GC content. There's some variation. Some of you said A, some of you said B. So here are actual isochore plots of those 2 species. Somebody help me figure out what we're looking at. We've got 2 plots. One of them is from each of those 2 species. One's from E. coli, and one's from Thermus aquaticus. I'm not saying that's necessarily the order they go in. Those are the 2 species. Which species goes with which plot? Who remembers what's on the x-axis of these plots? This one. X-axis represents gene sequence, DNA sequence. In this case, it's times 10 to the 5. So that means we've got position 20,000 base pairs in the DNA sequence of whatever organism this is, 40,000, 60,000, 80,000 base pairs. So we're talking about a genome that's about 100,000 letters long. What's its average percent GC based on this plot? About 0.5, about 50%. Okay. What about the other plot? What's its average percent GC? It's around maybe just a little under 70. So which plot goes with which species, do you think? Most of you guessed correctly about the Socrative quiz question. So who's got the higher GC content, TAC or E. coli? Thermus aquaticus. The organism that lives in the hotter environment, as we predicted last class when we discussed this, if you live in a hotter environment, you want more G's and C's in your genome because the DNA is more stable because more hydrogen bonds between G's and C's than A's and T's. So hot, H bonds. Any questions about this? Yes, sir.
So is this stronger because of the hydrogen bonds or because how it's placed?
There's actually some debate about what exactly makes the DNA stronger, the double strands more tightly bound. The default answer is it's the increased number of hydrogen bonds, but there are other factors that have to be considered about how the DNA is structured aside from that. So my cop-out answer is it's the number of hydrogen bonds, but it's almost certainly more complicated than that. Okay. So we're going to do our usual thing in class today. We've already been reviewing a little bit about last class. I'm going to do a tiny bit more. The Socrative poll we had at the end of the last class, you gave me really good feedback. I'm trying to incorporate your feedback on what it is that you feel comfortable with in class, what you're uncomfortable with so far. And this is what a lot of you broadly said you wanted more information about for this chapter and for the whole term as well. Why some mutations are deleterious, that is bad, and others aren't. We're going to cover that at the end of the term when we talk about central dogma. Polyploidy and why it's usually lethal. That is, if you've got too many chromosomes, why is that bad? Shouldn't that be, could it be beneficial? Got extra chromosomes, how could that possibly be bad? We'll talk about that in the next chapter. So not today, but next Wednesday when we come back together. Remember, don't come Monday, Labor Day. No one's going to be here other than you. Wanted to know more about topoisomerase. That's coming in the DNA replication chapter. Karyotyping, a few of you mentioned this term karyotyping and karyograms. What the hell is that, in nicer words? That's next chapter as well. The origin of replication we talk about in the DNA replication chapter. So there's a lot of this coming soon. I do want to calm some nerves. If I haven't gone into the detail you're expecting in class yet, at least I'm not going to put it on an exam until we get there. So do please ask questions when you're confused about something. If you have a question about terminology, if you want to know about how something applies, but I might say we're going to cover that more in detail later. So far in class, we've covered the amount of detail you need to know to succeed in the class. So here's what we're doing in chapter 10, sections 5 and 6. Today, these are the main points. The difference between an interphase chromosome and a metaphase chromosome. We're going to talk about chromatin structure and nucleosomes, because last class, a lot of you said these things at the bottom of the slide. You want to know about chromatin, chromosome compaction, what the hell is a nucleosome, how does it relate to chromatin, what are histones. So we're going to talk about all of this today. I'm a huge fan of analogies. You'll get used to this after a while, I think. So how do nucleosomes and chromatin relate to DNA? The nucleosomes are kind of like the curlers that you wrap the DNA in. So nucleosomes are made up of proteins that help compact DNA. There's a great picture in your textbook. I haven't put it up here for copyright reasons or whatever. It's in your textbook. Take a look at it. It shows what DNA looks like when you extract all the proteins from a chromosome. You take a chromosome, you chemically treat it to take all the proteins out, and what happens to the DNA? It no longer looks like a neatly packaged chromosome, something that we might see at metaphase that looks like that, it looks something like that. Right? Same idea here. Nucleosomes are these proteins. What are the proteins that make up nucleosomes called?
Histones.
Those are the histones. So a nucleosome, each single nucleosome is made up of 8 histone proteins. So you can think of that as one curler in this analogy. These 8 proteins, 8 histone proteins form one nucleosome. DNA wraps around that nucleosome or curler. And if you take all of those proteins out, if you don't have any proteins in your chromosomes, then your chromosomes look like hair when it's humid and not at all like my hair. If you've got curly hair and it's humid out, maybe it looks like this. It's the same process when you take the proteins out of DNA. So chromosomes, bottom line, have 2 components. There's the DNA, obviously, but chromosomes also are intimately associated with the incorporation of proteins. And you've probably seen this before, but these histones, I should have been an art major, form octamers. The 8 of them together form this regular shape that's what we call the nucleosome. And so DNA comes and wraps around and around those 8 proteins. And then it continues on to the next nucleosome where it's wrapped around those 8 histone proteins, one nucleosome, and so forth. So that's the primary level of DNA compaction, what our chromosomes really look like inside the cell. It's DNA wrapped around proteins, like hair and curls. Yeah?
Wouldn't it be more, I mean, why does it do that? 'Cause for me, I imagine if we get a piece of string and I put something in the middle, it takes up more space than if I just roll up the string. I mean, I know that the proteins are more basic, but they got that NH plus on it, so it binds to the negative charge.
Exactly. So great question, and you answered it yourself.
No, but I'm saying, like, but why can't the DNA just wrap around us? Is it because they're all negatively charged and they'll repel each other?
Right.
Okay.
So the question for those of you that might not have heard it was, why do we need proteins? If you have a piece of string, you just kind of like smash all the string together. It would take up less space if string is the DNA. It would take up less space than if you wrapped the string around some proteins. It would take up more space. But, as you noted, DNA is negatively charged and it repels itself. So you can't force DNA into a package smaller than when it's compacted with proteins. And it's because those proteins have positive charges that counteract the negative charges on the DNA backbone. Excellent. Modeling the scientific process. Ask a question, come up with some hypotheses for why it might or might not be true. Answered your own question. At the same time, you do have to start wondering, how much protein does each cell have to produce to wrap the billions of nucleotides we have in our genome up into chromatin in every one of our cells? A lot of protein. But we do it anyway. So there must be a good reason for it. Our cells wouldn't waste that much protein if there wasn't a benefit, supposedly.
What happens to the protein after DNA replication and then the DNA unwinds? What happens to the protein?
So short answer, we'll get to it in the DNA replication section, but no, you're fine. Long answer, briefly. I don't know. It's Friday. Happy Friday, everybody. So the histones temporarily become unassociated from the DNA during replication, and then as soon as the DNA is replicated, there are enzymes there that are sitting there that stick those histones back in and wrap the DNA back up. So there is a point in the cell cycle when your DNA temporarily isn't, parts of your DNA temporarily aren't associated with histones. Excellent question. Always good to be thinking about the exceptions, because remember, it's the exceptions where we learn something interesting about biology. Not when everything follows the rules and everything is as we expect. That's kind of boring. Now, 2 more things about chromatin. These are additional vocabulary items you've seen in the reading. Euchromatin versus heterochromatin. So remember, chromatin we're talking about is that combination of DNA plus protein. Euchromatin is sort of colloquially termed a more open DNA structure. Heterochromatin is commonly referred to as more compact. So there's a continuum in the cell, and different chromosomes could have different chromatin states. Different parts of different chromosomes can be euchromatin, and other parts of the same chromosome could be heterochromatin at the same time. And you might think about it like this. So if each one of these red circles is one nucleosome, 8 histones, sometimes the DNA and the histones might be loosely associated with each other like that. So the DNA generally is not tightly associated with proteins. There are all of these regions in the middle where the DNA isn't in contact with a protein. It's just hanging out there in the nucleus. On the other hand, there are times when chromatin is very compact, when all those nucleosomes are bunched up next to each other, and bunches of nucleosomes are bunched up with other bunches of nucleosomes, and that's what your reading talked about as sort of higher order chromatin structure. Beads on the string and supercoiling. So that there's still the same amount of DNA wrapped around each one of these histones, each nucleosome. But the DNA now, you can see, maybe, with my horrible drawing, is much more tightly associated with proteins. There isn't this linker region of DNA that's not associated with proteins between the nucleosomes. So these define, to an extent, what euchromatin looks like versus what heterochromatin looks like. Questions? Yeah?
So is this how it's determined which genes are expressed in?
This, which we will see when we talk about gene expression, you're going to hate me for this. This is my style, but yes, absolutely. Good forward-thinking question. Why does this matter? Why are we bothering spending time in class talking about how our DNA is packaged? Partly it's because if you have a gene in here, how is it going to be turned on? How is that gene going to be read by proteins and transcribed into RNA? It's easy. So you might say the genes that are in heterochromatin are turned off. They're never read and transcribed and translated, whereas genes that are in euchromatin are generally thought of to be on. They're because the DNA is exposed in the nucleus, other proteins can come in and transcribe and translate the genes and produce proteins. We go into more detail about that later in the semester, but fabulous question. That is the main reason we care about euchromatin versus heterochromatin. Okay. Other questions? Here's an example of differences in chromatin structure. I mentioned briefly you could have a chromosome where part of it's euchromatin and part of it is heterochromatin. Here's a metaphase chromosome spread of a human female. And in this case, the scientists that have produced this image have treated these chromosomes with a chemical that creates what's called here C-bands. So these are C-banded chromosomes. And it's these dark spots that are the C-bands. They've reacted positively with whatever chemical was put on the chromosomes, and that's why the chromosomes are darker there. So you add a chemical, this chemical identifies what we call C-bands. Where are the C-bands if you describe where they're located on the chromosomes as you see them? They look like they're at the centromeres. Guess why they're called C-bands? So this is a technique that cytogeneticists, doctors that study chromosomes used to identify the centromeres of chromosomes. If you want to know where the centromere is, apply this particular chemical, look at the chromosomes, you see easily where the centromeres are. The reason that this staining technique works is because the centromeres are heterochromatic. Centromeres of chromosomes are those, or parts, every chromosome has that tightly compacted heterochromatin nucleosome structure. And that's what we're seeing here. Those dark bands are really just the chemical identifying where are the nucleosomes. Nucleosomes are more closely associated, they're denser at the centromeres, so it appears darker when we look at the picture of the chromosome.
Dr. Oz?
Yeah?
Does that mean that all the heterochromatin is the junk DNA because the Euchromatin is the one that gets expressed, so it has to be longer?
So there's a question about, is there an association between repetitive DNA and where your heterochromatin is? That's a difficult question to answer. In general, that junk DNA, repetitive DNA sequences are spread throughout all of our chromosomes. So we're not seeing evidence of that in this particular picture. But our centromeres are repetitive DNA. So after you zoom in on the chromosomes and you actually look at the DNA sequence of human centromeres, it's like 181 base pairs, I think, repeat over and over and over and over again. So in this particular case, these centromeres are heterochromatin, tightly compacted, in part because they're repetitive. That's not the rule, though. That's just a specific observation for the case of centromeres. Are there any dark blobs up here that are not at centromeres? So if you do see any dark, let's see, who said that? Help me out. So where on this do you see dark? Not okay. So yeah, you've got some random little maybe fragments of DNA. I don't know what the heck those dark blobs are that don't look like they're associated with chromosomes. We have some other regions like this one here, where there's a little bit darker band. It's not nearly as black as the centromeres, but it's pretty close. So those might, in answer to the same question, those might be regions, the darker regions that aren't at centromeres could be spots in the genome where there are repetitive DNA elements. So this technique might not be picking up just the heterochromatin at centromeres, but other regions of the genome where the chromatin is highly compacted, heterochromatin. So within a single chromosome, bottom line, you can have some regions that are euchromatic, open, and some that are heterochromatic, compacted structure, like centromeres. Okay. A lot of you wanted to know about how the cell cycle and DNA replication impacts chromatin structure. There's a lot of the questions I got at the Socratic quiz, the end of last class. So here's a simple diagram of the cell cycle. What happens at M? Mitosis. Okay, so that's cell division. So right after mitosis, let's say we're talking about humans, just for example, because we're all pretty familiar with this. How many copies of every chromosome will be in each of the two cells that are produced from mitosis? How many copies of every chromosome? We're diploid, so we've got 2 copies of every chromosome. So I'll just diagram those as vertical sticks. What happens during G1 phase? Does anything happen to the DNA during growth phase? When's the first step in the cell cycle when something happens to the DNA? It's one of these 4 stages. It's not G1, that's just growth. What's S stand for? Synthesis of DNA, okay. So take one of those 2 daughter cells. early in S phase. S phase has just started. Nothing is replicated yet. But what happens to the chromosomes in S phase? They start to do something. Most of the cell cycle in growth phase, our genes are doing things. They're being transcribed and translated and turned into proteins. So it's in the growth phase when transcription and translation happen. TXN and TLN, shorthand for transcription and translation. But during S phase, our chromosomes are no longer euchromatic. They become heterochromatic. They become heterochromatic. Our chromosomes condense. Then after synthesis, we have 2 copies of every chromosome. They decondense. They become euchromatic again, because in the second stage of growth, G2, once again, genes are being transcribed, translated, turned into proteins. And then in mitosis, the chromosomes segregate into the daughter cells. So there's a pattern of euchromatin in growth phases, compression, compaction, heterochromatinization in synthesis, and then relaxation of that condensation during G2. And in mitosis is when they have to compact. So it's not necessarily the case that these are as heterochromatic. There's some debate about this. But it's definitely when you start to actually move the chromosomes around and think to yourselves, why is this important? It's when you actually have a cell trying to physically move and separate the chromosomes that chromatin structure is really important, that you're heterochromatic.
So when we have a DNA replication, wouldn't you want your DNA to be unwind so it'd be easier for proteins to come in than heterochromatic?
That's right. So that's why there's some, it depends on the organism you're looking at. So there's some cases where that might not be the case, but yes, absolutely in general, during synthesis, you expect Euchromatin. At some point, and this is again don't get bogged down in this detail necessarily, but this is where you might expect some regions of the chromosome are open, euchromatic for replication, but then become heterochromatinized again. How many in here have heard about a Barr body? Can I have a show of hands? Barr body? This is something that happens to a few of you. This is something that happens to X chromosomes in females. So guys, you can relax. This process doesn't happen to you. But if you're a female, you have 2 X chromosomes. But in every one of your cells, one of those X chromosomes has been turned off or silenced. It has become permanently heterochromatic. And the reason that this occurs is because we have females and we have males. We guys, we've only got 1 X chromosome. Take pity on us. So we've got males only have 1 X chromosome. And this is why, ladies, one of your X chromosomes gets shut off. Is because we've only got 1 X chromosome. And so to make it fair, this is, of course, not a scientific argument. To make it fair to us, you turn off one of your X chromosomes during development. So that you've got 1 X chromosome, we've got 1 X chromosome. Everybody's happy because it's equitable. That's the storytelling version of it. We might get into more details later about how this happens and why this happens. So this is an example of where you have a chromosome that's completely heterochromatic. All of the DNA in that inactive X chromosome, the Barr body, is as tightly wound as it can get. But you still have to replicate that chromosome every time you do cell division. So that whole chromosome has to become euchromatic and then get packaged again into a Barr body. really briefly, one or many of you asked about the difference between interphase chromosomes and metaphase chromosomes. Last class, I showed you the same picture. And I said something really stupid and non-scientific about this thing that I've pointed at with the blue arrow here looking like a fuzzy brain or something like that. I don't remember what exactly I said. What is this? I asked you what is this last class, but I didn't follow up on that question. So we've got a lot of individual chromosomes we can see. What's that blob? I'll give you a hint. It's also DNA. Is this DNA that's at interphase or metaphase? So all of these chromosomes, when you actually see individual chromosomes when they look like what we would normally call chromosomes, are metaphase. This blob is an interphase nucleus. So that's exactly the same amount of DNA as shown there in all of those individual chromosomes at metaphase. It's just that that's the entire nucleus. That's all of the DNA in the cell, but it's all unwound. It's euchromatic. It's filling up the entire nucleus. It's disorganized in a sense. So we have one entire nucleus. That's all of these chromosomes that are at metaphase, but packed into a nice, neat little nucleus package. So that is the difference between interphase and metaphase. During interphase, chromosomes are all strung out, in a manner of speaking, and unwound, but they really highly condense during metaphase, right before segregation of chromosomes into daughter cells occurs. I'm going to skip this analogy. And so you should be comfortable. It's a good analogy, but we don't have time for a bunch of analogies. You should be comfortable at this point connecting a term from the left side of the screen to the term at the right side. Do you normally see Euchromatin, open DNA, during interphase or metaphase? So Euchromatin when the DNA is all interphase. Yep. And it's during metaphase, when the chromosome is really compact, we see them as individual chromosomes. We see them as individual chromosomes because they're tightly compacted. Metaphase, heterochromatin. We're going to take a little bit of time and work on this exercise that I asked everybody to do for today, or at least to attempt to do. So I want to form small groups. Organize yourselves with your neighbors into groups of 3 to 5. Hopefully at least one of you did this exercise. That's the goal. So if none of you did, make a slightly bigger group until you find and absorb somebody who did this. So the idea was to find a picture of a chromosome and to draw how you think it's related to a double helix. So quickly, 3 minutes or so, get into your groups, share your images with each other just by looking at your tablets. Pick the best representative from your group and attach it to this Google Classroom assignment.
You appreciate
Okay. I'm just trying to think if there's anything else in how this data might be here involving. It looks like the troubleshooting is pretty minimal. So it should be okay. Yeah, but thanks for definitely no, you're totally fine. Yeah. Yep. One's
Like, there are different phases in the cell cycle.
so that's always the first response. So they must be at different stages of replication, for example. But, and this is really, really important for those of you in the back especially. Anytime you see a chromosome that's condensed, that looks like what we call a chromosome, what stage in the cell cycle is it in? These are all metaphase chromosomes. So all of these pictures of chromosomes that you're looking at, when you see a thing that is a chromosome, it's at metaphase. So if these are all human metaphase, all of these pictures of human chromosomes are all at metaphase. Why are they different looking? Thoughts?
So, the
For example, these two things. 2 copies of human chromosome 1. Each of those things that looks like a single object is 2 double helices. So I do get a lot of questions often about some people finding, students included, finding images of chromosomes that look like that, the X's that we're maybe familiar with, and others finding images that look like that, where the chromosomes don't look like X's, and saying, what's the difference? So I wanted, hopefully I haven't made the problem worse, wanted to clarify the distinction. All of these are metaphase chromosomes. It's just that some are earlier and some are later in that stage than others. So this is not vocabulary that I'm interested in you memorizing, but this did show up in this chapter. There's this protein called condensin. Oh dear, I need to turn off notifications. I'm glad to see that people are actually making comments during class though, and I should check my Twitter feed too. So there's this protein called condensin. And condensin, I'm going to show here in black, holds the 2 newly replicated chromosomes together during the early part of synthesis, and sorry, not synthesis, mitosis, the M phase in the cell cycle. So this is early S, just after synthesis, right before cell division, mitosis. Later in mitosis, that condensin is cut. Dare I draw scissors? I don't know, I'm not that much of a, let's see, scissors. No, that doesn't look like scissors at all. Let's see. Now, I don't know what that is. Anyway, there's an enzyme that's, so cohesin, right? Cohesive, sticky. Cohesin is a protein that sticks the chromosomes together. And then separase, the protein, comes and cuts the cohesin, or condensin and cohesin. There are two proteins that are involved in that process, condensin and cohesin. So separase comes along and clips all of the cohesin that sticks the two chromosomes together. So it is a cell cycle thing, the reason that those chromosomes look different. Some of them look like a single stick, and some of them look like two sticks that are crossing each other. Same thing. Both of those are two double helices side by side. It's just that early, at step 1, like I'm drawing here, these two double helices are right next to each other physically. They're laying side by side. So you don't see any separation between the two chromosomes. They look like, but later in the cell cycle, after separase has separated the arms of the chromosomes, then they start to lay out in that more familiar X-shaped structure. Still, each of those objects is a single double helix, like many of you drew in this exercise.
Dr. Ross,
Yeah.
they don't have that, the cohesion in the middle, they won't have the X shape, but if they don't have it, it'll just look like the linear chromosome?
So all of the chromosomes start out looking like this. So they'll all start earlier in the cell cycle looking like that, like one chromosome stick shape, as was in some of those Google Images results. It's later in the cell cycle, after separase is turned on, that those two replicated chromosomes can spread apart. And that's when you start getting things that look like this X-shaped chromosome. And the point of doing this was, okay. So for the last couple of minutes of class, we'll do one more group exercise. This will set us up nicely for our next class meeting on Wednesday. So in the next minute, if you can, count how many chromosomes are in this image. Once you do, talk to your neighbor briefly. See if you can figure out how to interpret how many chromosomes are here. Feel free to talk. We'll come back together in 1 minute and do a wrap-up. Okay, so really quickly, I know some of you are still working on it, that's fine. If you're done, how many chromosomes did you count? 42, 46, 41, 43. What's one thing you learned about doing this in 1 minute in class? It's hard sometimes to do this. So how many chromosomes is that? I think that's, yeah. I say 3, someone else says 2. So this is a real problem in cytogenetics when you look at a chromosome spread. Sometimes the chromosomes don't lie down nicely, separated from each other like we'd like. So the question to think about for next time, and then I want to show you the slide that shows what to do for next time, then we're done, I promise. So do, after class, go through and very carefully count how many chromosomes you think are in this image. it does wind up being an odd number. So that should be the answer you wind up getting. Try to think, as you read the material for next class, why do you see an odd number of chromosomes in this metaphase chromosome spread? That's the question. So next time, there's a reading due Wednesday. I've also made a pre-lecture video to watch. It's on YouTube. It's about 20 minutes. Please watch that in advance of class on Wednesday as well. So one reading assignment, one movie to watch on YouTube. I'll see you on Wednesday. Yes. Have a good 3-day weekend.